So we're going to be giving a basic intro to the DNA structure. So here we have a basic nucleic acid, right? So the first thing we want to note is that there's different parts to it. So individually we have the sugar portion, we have the base portion, and the phosphate portion. So individually, just this sugar portion is called deoxyribose. Similarly, if we had something like this would be ribose. And we'll see later that this deoxyribose is what makes DNA, and this ribose is what makes RNA. So those are the only difference really, along with a couple others, between DNA and RNA. But back to this, if we included the base with this sugar, we would have something called deoxynucleoside. Okay, so remember this last ending, the side part. If we include everything, well, now this is the deoxynucleotide. So tide, remember that ending. So nucleoside versus a nucleotide. Nucleotide includes the phosphate, nucleoside doesn't. Okay, so what are these bases? These bases can be a couple of things. They can be A, T, G, C, and in the case of RNA, they can be U. Okay. So for purines, these would be the A and the G, adenine and guanine. And then for pyrimidines, they are C, U, and T. Okay? So one way to remember this is pyramids cut. Okay? So pyramids cut sounds kind of corny, but pyramids, pyrimidines, cut, CUT, you'll memorize it for sure. You'll have no problem because everything else is just A and G, and you know those have to be purines if they're not pyrimidines. Alright? So now that we have that down, we need to go into how to make a phosphodiester bond. And the phosphodiester bond is how you link multiple nucleic acids together. Okay? So if we have individually, we have two nucleic acids right here, basic structure, nothing special to them. But if we have two nucleic acids right there, how can we form this phosphodiester bond? So what happens is, this is a hydrolysis reaction. So this little right angle right there, that's also a CH2. We remember from OChem, anything that's just a point is going to be a carbon right there. So this hydroxy group comes in and attacks the carbon and releases the phosphate group. Okay? So once that's released, then we just form this bond. And what was released is H2O. So one thing they may ask is, what type of reaction is this? And this is hydrolysis. Or a dehydration reaction is another way they would call it. So we see that we release the water in the process. Okay, so the next thing we're going to be looking at is the Watson-Crick Model and Chargaff's rule. Okay, so Watson-Crick Model. So Watson and Crick, those are the guys who invented it. Not important. What's important is what they actually found. So they found three things that we really need to know. One is that DNA is anti-parallel. So what does that mean? So in one direction you have DNA going 5' to 3', and in another direction we have 5' to 3'. Right? So they're opposite, and that's why they're that's why they're anti-parallel. And so to add on to that, they're in a double helix. So we always see that they're always paired. And this is only for DNA. This is only strictly for DNA because double helix is not relevant for RNA. We'll see that later. But DNA is double helix. I can't really draw it, but you can imagine it wraps around. It wraps around like a double helix. And so they're always paired together. They never come singly. And the third one is A bonds with T and G bonds with C. And A to T is 2 hydrogen bonds, and G to C is 3 hydrogen bonds. And we saw A, T, G, C, and U as the bases before. People have asked me, do we have to memorize those bases? Do we have to memorize how they look? The answer is no. Don't worry about memorizing everything. Just know which one is which one's a pyrimid, which one's a pyrimidine. So that's A, T, G, and C, 2 hydrogen bonds, 3 hydrogen bonds, and if you actually looked at the structures you would see why, but it's not too important, just memorize 2 and 3. and Chargaff's rule. So this relates to this ATGC pair. So Chargaff's rule says a couple of things. So A equals T. So the concentration of A equals the concentration of T. And that should make sense because A always bonds with T. If this wasn't the case, if these two weren't together, then you have some type of mutation. So G equals C, same thing. And A plus G equals T plus C. Okay, so this should make sense. Just purines equals pyrimidines. The concentration of the two equal each other. Okay, so they could easily give us a question where they asked us, if they gave us a percentage of one of them. So let's say they gave that A was 20%. Then they may ask us, what is the percentage of something else? So what is the percentage of G or C? So let's just say, what is the percentage of G, the concentration of G? Well, we could figure this out, okay? So we know that A is 20, so let's plug it into this equation here. And all this equation says is that we know that the total percentage of A, T, G, and C has to equal 100. Right? If this is DNA, it has to equal 100 because those are the only 4 nucleotides that we have. Okay? So we know A is 20. T, what is T? Well, we know T has to also equal to A, so T will also be 20. And we want to find G, so that's the unknown. But also C, we know C has to equal to G. So we can solve it like that. G plus G equals 100 minus 40. 2G equals 60 and G equals 30. Alright, so fairly simple. This is all we really need to figure out for the MCAT in terms of this DNA. So that's all the math that we'll see. Alright, so in terms of packaging, and this is specific for eukaryotes because prokaryotes package in a different way. And they just do a different type of supercoating, but we'll look at only eukaryote supercoating. Okay, so supercoating is what it's called for packaging for eukaryotes. So what happens is we have these proteins, so these histones, and these are proteins where DNA can wrap around. So if you can imagine DNA wrap around like that, and you may have learned in your genetics classes that there's a certain number of turns that it wraps around, and there's a certain number of histones in one block. Is that important for the MCAT? Probably not. You don't need to worry about those, so I'm just going to go very basic. So DNA wraps around histones. This will equal something called a nucleosome. Okay, now nucleosomes can wrap around together. So imagine nucleosomes wrapping around together. Then you'll get chromatin. Chromatin wrapping around together, you'll get a chromosome. And so remember, chromosomes typically look something like the little X like that. So that's pretty much chromosome packaging. So we talked about DNA, but what is the reason? Why do we even have DNA? Well, DNA is the genetic building block. So the genetic code. It's what makes us. But really, what's most important is the central dogma. And what the central dogma says is DNA goes to RNA, and RNA goes to protein. So ultimately, what's the point of DNA? Well, the ultimate goal is protein. It's to produce protein. And proteins are used in enzymes. They're used to make everything pretty much, to make us, to get everything to function properly. So proteins is really what makes us. We need DNA in order to make us what we are, and that's pretty much the main purpose, is this central dogma. So never forget this: DNA to RNA to protein, everything does it, whether or not you're a plant, an animal, bacteria. One thing that may not do it, that doesn't follow the central dogma, are viruses. And we'll see those later on, that they can go in the reverse direction. Retroviruses can do that, but don't worry about that. Just know that central dogma, always DNA, RNA, protein.